Chapter 8 Cell, the unit of life. Tools of cytology. Microscopy. Common unit of measurement in microscopy as well as cytology is nanometer. 1 nanometer is 10 raised to 10 raised to minus 9 meter. 1 nanometer is 10 angstrom. The unit of measurement of cell size is micrometers. 1 micrometer is 10 raised to minus 6 meter. Leeuwenhoek is called as the father of microscopy. Ability to distinguish two closely placed objects is called the resolving power or the limit of resolution. Resolving power of human eye is 100 micrometers or 0.1 millimeter. It means human eye cannot see objects less than 100 micrometer in size. Resolving limit of our eyes is 1 by 60th 420 micrometers. Resolving power of the transmission electron microscopes is 0.2 nanometer, 2 angstrom. Dissolving power of the electron microscope is 10 angstrom. Resolving power of scanning electron microscope SEM is 5 to 10 nanometer. Resolving power of light microscope is 0.2 micrometer or 0.002 millimeters or 0.25 micrometers. Resolving power of light microscope is 3000 angstrom. The electron microscope has a high resolution power. This is due to the very low wavelength of electron beam. Electron used in the electron microscopes have a wavelength of 0.5 angstrom. The resolving power of a compound microscope will enhance with decrease in wavelength of light and increase in numerical aperture. Resolving power is calculated by Abbe equation. Value of RP is 0.16 by lambda by Na. Na is numerical aperture. Highest resolving power is obtained with a short wavelength. Low is the value of resolution, highest is the resolving power. Compound microscope or light microscope. In compound microscopes, three types of lenses are used: ocular, objective, and condenser. Magnification of a compound microscope is a product of the ocular lens eyepiece and the objectives. M is equal to OC into OP. Magnification of a compound microscope is not related with numerical aperture. In compound microscopes, the image form is erect and vertical. Phase contrast microscope, developed by Janikar. Phase contrast microscope is useful for the studying of living cells without staining. It is used to study the dividing cells. A scientist wants to study a metaphasic behavior of chromosomes in a living cell. He must use phase contrast microscope. Differential interface contrast microscope. It is used for the determination of dry weight of macromolecules like RNA, DNA, and proteins. Fluorescent microscope. In fluorescent microscope, the components are visualized through the coating and fluorochrome. Fluorochrome are the chemical substances which emits visible light on irradiations with UV rays. High wavelength of UV rays are used in fluorescence microscope. In this microscope, filter for UV radiation is provided for protecting eyes. Distribution of two or more specific molecules within a cell is studied by fluorescent microscope. Fluidity of membrane is observed through fluorescence microscope. Electron microscope invented by Knoll and Ruska. For the invention of electron microscopes, Ernest Ruska was awarded Nobel Prize in Physics in 1986. Electron microscopes have objectives and ocular lenses. Light source in electron microscopes is beam of electrons. Electron microscopes is based on three electromagnetic theory. A major breakthrough in the studies of cells came with the development of electron microscope. This is because the resolution power of the electron microscope is much higher than that of light microscope. Electron microscope is more advantageous than light microscope because it has higher magnification. Ultrastructure of cell organ can be studied best through the EM. EM is used for viewing cell structure. EM has revealed the occurrence of ribosomes. Depth of the object can be studied through scanning EM. Centrifugation technique introduced by Svedberg, for which they awarded the Nobel Prize. Centrifugation technique is based on density. High speed centrifugation is called ultracentrifugation. Pure fractions of cellular components is isolated by differential centrifugation. Ultracentrifugation is employed by separating proteins from other fraction. Subcellular components are separated with the help of centrifugation fractionation. Ultracentrifugation is a common method to determine weight of proteins. Intact chloroplasts of green cells can be isolated with the help of sugar solution. Lighter and heavier forms of an elements can be separated by buoyant density centrifugation technique. Autoradiography. This is called as a tracer or the radio tracer technique. Tracer elements are radioisotopes. Tracing the various components of the cell by using the tracer element radioisotopes or radioactive isotopes is called as autoradiography. By autoradiography, the amount of DNA, RNA, and protein can be known at any times. DNA synthesis can be measured by estimating the incorporation of radio-labeled thymidine. Radio tracer technique does not provide any proof about the number of helix in the DNA. Uridine is labeled by incorporating H3 for studying RNA synthesis. N15 and O18 are not radioactive isotopes, but they are heavy isotopes. P32, H3, C4, S35 are radioactive isotopes. I30, 131. X-ray diffraction. Arrangement of atoms and molecular groups in DNA and RNA can be studied through X-ray diffraction. Binding of specific proteins on regulatory DNA sequences can be studied by the means of X-ray crystallography. Fifth, electrophoresis. Particles of the different charges and sizes are separated under the influence of electric field. Electrophoresis can help separate proteins, amino acids, nucleic acids, and nucleotides. Cytochemistry staining. Certain dyes are used in cell of living cells or materials. They are called as vital stains. Neutral red, Golgi complex, methylene blue, critical Gb, Janus green, mitochondria. Dyes may be acidic, basic, or neutral. Acidic dyes can be of cytoplasmic structure, Janus green, methylene blue. Basic dyes stains the chromosome and other structures, crystal violet, basic fuchsin, and sapphire. Fusels or shifts reactions was developed by Fusion and Rosenberg. It employs basic fuchsin and After acid hydrolysis, this reaction is specific for DNA. Fusion stain for DNA is basic fusion. Fusion reactions involves removal of purines by hydrolysis. Chromosomes or nuclei is also stained by cadmium or acetocarmine or orcine. Saffirin is used to stain lignified cell wall. Middle lamella calcium pancreate is stained by methylene blue. Iodine is used to stain starch. Blue color iodine test. Iodine test detects starch for carbohydrate. Sudan black B is used to stain lipid. Metachromasia is the characteristic change in the color of staining. Example, toluidine the becomes toluidine blue becomes pink when both bound with cartilage. Cell, the unit of life. Study of cell structure is called cytology. Study of cell structure and function and reproduction is called cell biology. When you look around, you see both living and non-living things. You must have wondered and asked yourselves that what is makes an organism living, or what is it that inanimate thing which thing does not have, which a living thing has. The answer to this is the presence of the basic unit of life, the cell in all living organisms. All living organisms are composed of cells. Some are composed of single cells and are called unicellular organisms, while the others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular or acellular, amoeba, yeast, etc. organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cells are fundamental structural and functional units of all living organisms. Cells was first discovered by Robert Hooke in 1665. Actually, cell wall of bottle cork. The term cellulae was given by Robert Hooke. Robert Hooke is the father of cytology. Hooke published his work in the book Micrographia. C.P. Swanson, father of modern cytology. Anton von Leeuwenhoek first saw the described live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. Living things are made up of cells, and this was stated by Lamarck. In 1838, Matthias Leden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant. Hypothesis. At the end of same time, Theodore Swan, a British zoologist, studied the different types of animal cells and reported that cells had a thinner outer layer which today is known as the plasma membrane. Plasma membrane or cell membrane was discovered by Swan, but term cell membrane was given by Najeli and Kramer. Term plasma lamella was given by Plough. Swan also concluded, based on his studies on plant tissues, that presence of cell wall is a unique character of the plant cells. Swan discovered the animal cells lacked cell wall. On the basis of this, Swan proposed hypothesis that the bodies of animals and plants are composed of the cells and the products of cells. Cell hypothesis. Schleiden and Swan together formulated the cell theory. This theory, however, did not explain how new cells were formed. Rudolf Virchow first explained that cell division and new cells are formed from pre-existing cells, omnicellula acellular, also called law of lineage or common ancestry. Louis Pasteur in 1862 proved that life arises from pre-existing life, genetics and evolution. He modified the hypothesis of Schleiden and Swan to give the direct theory of final shape. Cell theory is understood today as all living organisms are composed of cells and cell products of cell. Cells are the fundamental structural and unit of organisms. All cells arise from pre-existing cell. Viruses are exception to cell theory as they do not have cellular organization. Dutrochet first stated that an individual organism has different types of cell. Capability of plant cell to form complete plants on the plant is called cellular totipotency. The ability of cell to form many phenotypes is called pluripotency. In tissue culture, embryoids are formed from pollen grains due to cellular totipotency. An overview of cell. We have earlier observed that onion peel and human cheek under cells under the microscope. The onion cell, which is a typical plant cell, has a distinct cell wall and its outer boundary, and just within it is a cell membrane. The cells of the human cheek have an outer membrane as a delimiting structure of the cell. The living substance of the cell is called protoplasm, given by Purkinje and Van Moll. The term sarcode was used for the living substances of cell by Dujardin. Protoplasm is the physical basis of life. This is called by Huxley. Protoplasm is complex or colloidal solution and forms 95% of the total weight of the body. Protoplasm is divided into two parts. The central part is called the nucleus and the peripheral extra. Nucleus part is called cytoplasm, Strasbourg. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome which in turn contains the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of cell. The cytoplasm is the main arena of the cellular activities in both plant and animal cells. Different cells of organelles are embedded in cytoplasm. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants, and mitochondria and contraphia. So ribosome is called as an organelle within an organelle. Animal cells contain another non-membrane bound organelle called as a centriole, which helps in cell division. Cell size and shape. Cells differs greatly in size, shape, and activities. Cell sizes are expressed in micrometers. For example, mycoplasmas are the smallest cells and are only 0.3 micrometer in length, while bacteria could be to 3 to 5 micrometers. Viruses are smaller, 0.02 to 0.2 micrometers than mycoplasma, but they do not have a cellular structure. The largest isolated single cell is that of an egg of an ostrich, 15 centimeter. Largest one cell living organisms is Acetabularia algae umbrella plant, 10 centimeter long. Okay, this diagram is very important. A typical eukaryotic cell is 10 to 20 micrometers. A typical bacteria 1 to 2 micrometer. PPL was 0.1 micrometer. Virus is 0.02 to 0.2 micrometers. So typically viruses are among the smallest. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Longest cells of the human body are nerve cells up to 90 centimeter in length. Nerve cells are branched to long to conduct impulses. Longest cell in the plant is phloem fiber or bast fiber over 1 meter in hemp. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the functions they perform. RBCs are round and biconcave to pass through capillaries and carry more oxygen. WBCs are amoeboid to do phagocytosis and diabetes. Columnar epithelial cells are long as to narrow. Mesophyll cells are round and oval. Tracheids are elongated to translocate water along to long distances. Smaller cells have higher surface area and volume ratio than larger cells. Smaller cells are metabolically more active as there is more area available for the exchange of materials between the cells. Smaller cells also has higher nuclear cytoplasmic ratio, that is ratio of nucleus to cytoplasm. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, pleuropneumonia-like organisms, and belongs to kingdom Monera. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. The organization of a prokaryotic cell is fundamentally similar, even though prokaryotics exhibit a wide variety of shapes and functions. All prokaryotic cells have a cell wall surrounding the cell membrane. The fluid matrix filling the cell is called the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have smaller circular DNA outside the genomic DNA. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have DNA outside the genomic DNA. These smaller DNA are called as plasmids, extra chromosomal material. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. The plasmid DNA is used to monitor bacterial transformation with foreign DNA. Plasmids were discovered by Hayes and Landerberg. When plasmid is associated with nucleoid or single chromosomes or genomic DNA of bacteria, it is called as episome. Chromosomes in a bacteria is always circular. Genophore term was coined by Hans This for bacterial chromosome. Nuclear membrane is found in eukaryotes. No organelles like the one in eukaryotes are found in the prokaryotic cells except for the ribosomes. Prokaryotes have something unique in the form of inclusions. DNA of E. coli is double-stranded and circular. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cells called cell envelope. Glycocalyx is the outermost covering of protoplasm of bacterial cell. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act as together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by the gram, namely those who take up the gram stain are gram positive and the others that do not take are gram negative bacteria. Glycocalyx differs in composition and thickness amongst different bacteria. It could be a loose sheet called the slime layer in some, while in others it may be thick and called as the capsule. Capsule allows the bacterium to hide from host immune system. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Cell wall presents between glycocalyx and plasma membrane. The plasma membrane is a semi-permeable in nature and interacts with the outside world. The membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell of the prokaryotes. They are essentially infoldings of the cell membrane. Mesosomes are functionally similar to mitochondria. These extensions are in the form of vesicles, tubules, and lamellae. Mesosomes helps in cell wall formation, DNA replication, and distribution to daughter cells. During replication in bacteria, DNA is anchored to the mesosome. Mesosome also helps in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like the cyanobacteria, there are other membranous extensions into the cytoplasm called as chromatophores, which contains pigments. Term chromatophores was given by Smith. Flagella. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in number and arrangement of flagella. Bacterial flagellum is composed of three parts: filaments, hooks, and basal body. Hook connect filaments and basal body. Hook is the thickest part of the flagellum. The filament is the longest portion and extends from the cell surface to the outside. Filament is made up of protein called flagellin. Besides flagella, pili and fimbriae, also surface structures from the bacteria, do not play a role in motility. The pili are long, elongated tubular structures made up of special proteins. The fimbriae are small bristle-like fibers sprouting out of the cell. Fimbriae are hairy appendages on certain bacterial wall. In some bacteria, they are known to help attach to the bacteria to rocks in the streams, and they also to host the tissue. Ribosomes. Ribosomes are membrane-less ribonuclear proteins particles. In prokaryotics, ribosomes are associated with plasma membrane of the cell. Bacterial protoplasm is granular due to ribosomes. They are 15 nanometer by 20 nanometer in the size and are made up of two subunits, 50S and 30S units, which then represent together 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosomes of a polysome translate mRNA into proteins. Inclusion bodies. These are non-living structures present in cytoplasm. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and lies free in the cytoplasm. Example: phosphate granules, cyanophycean granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple photosynthetic bacteria. Eukaryotic cells, except Monera, other four kingdoms possess eukaryotic organization. The eukaryotic includes all protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, the eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. The genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plants and animal cells are different as the former possess cell walls, plastids, and the last central vacuoles, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. 1. Cell wall. Cell wall is present in plant, fungi, and some protists, chrysophytes, dinoflagellates, but absent in animals and some protists, euglena and protozoa. A non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and plants. Gametes of both plant and animals do not have cell wall. Structural elements of cell wall is microfibrils. Cell wall is not only gives shapes to the cell and protects them from mechanical damage, infections. It also helps in the cell-to-cell interaction and provides barrier to undesirable macromolecules. Cell wall shows complete permeability. Cell wall is freely permeable. Algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. Very important. Cell wall can have up to 3 layers: primary wall, secondary wall, and sometimes tertiary wall. Primary wall is the first form wall of the cell. The cell wall of the young plants, the cell primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side towards membrane side of the cell. Secondary wall is produced in some mature cells on the inner side of primary wall. In some gymnosperms, the 30 wall is also laid down, which is formed of xylem. So primary wall is the outermost layer of the cell wall. Secondary wall may be of the deposition of lignin. Primary wall is elastic due to absence of lignin. Pit is formed in cell wall due to lack of secondary wall material. The middle lamella is a layer of mainly of calcium pectate, which holds or glues the different neighboring cells together. Middle lamella is made up of calcium pectate mainly and magnesium pectate. During ripening of fruit becomes soft and pulpy due to dissolution of middle lamella. The cell wall in the middle lamella may be transferred by plasmodesmata, which connects the cytoplasm of neighboring cells. Plasmodesmata are the protoplasmic stranded between adjacent plant cells. Plasmodesmata were discovered and named by Strasburger. Plasmodesmata connection helps in movement of substances between cells. Welded areas between the two adjacent animals are called as desmosomes. Desmosomes is modification of plasma membrane. Desmosomes are not found in prokaryotic cells. A cell without cell wall is called as protoplast. Cell membrane. Second is the cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to reduce the possible structure of the plasma membrane. These studies showed that the cell membrane is composed of the lipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of the saturated hydrocarbons is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglycerides. Later, the biochemical investigation clearly revealed that the cell membranes also possess proteins and carbohydrates. The ratio of protein and lipids vary considerably and different in cell types. In human beings, the membrane of the erythrocytes has approximately 52% of the protein and 40% of the lipids. Cell membrane is composed of lipoprotein. Membrane surrounding an organelle is called a subcellular membrane. Subcellular membrane are absent in the prokaryotic cells. In a cell, most abundant membrane is the membrane of ER. Many types of models are given to explain the structure of the cell membrane. One, Daniel and Davison model. They proposed first lamella model in 1935, in which they proposed that cell membrane contains 4 layers, 2 of phosphorus and 2 of proteins. Robertson modified the model of Danieli and Davison, a lipid bilayer being sandwiched between 2 layers of proteins on either side of it. Thickness of membrane is about 75 angstrom, with a central lipid layer of 35 angstrom and 2 peripheral protein layers of 20 angstrom each. Robertson gives the correct concept of unit membrane, in which the correct sequence of protein L and P in the cell membrane is L, P, P, L, L, P. Lipid molecules of the cell membrane are arranged parallelly. Phospholipid molecules of cell membrane possess one polar head and non-two polar heads. Okay, so this is the most important fluid mosaic model. An improved model of the structure of cell membrane was proposed by Singer and Nicholson and widely accepted as the fluid mosaic model. This is the latest model for plasma membrane. According to this model, membrane does not have a uniform arrangement of lipids and proteins, but is a mosaic of two. Protein molecules are present both inside and on the outside side of the lipid bilayer. Depending on the ease of extraction, membrane proteins can be classified as integral or peripheral. Integral proteins are called as intrinsic or intergranule proteins, while the external are called as extrinsic or peripheral proteins. Peripheral proteins lie on the surface of membrane, while the integral proteins are partially or totally buried in the cell membrane. According to fluid mosaic model, plasma membrane is composed of phospholipids, extrinsic proteins, and intrinsic proteins. According to this, the quasi-fluid nature of the lipids enables lateral movement of proteins within the overall bilayer. The membrane is quasi-fluid in nature. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important for the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division, etc. Certain membrane proteins act as hormone receptors. Cell membrane causes compartmentalization in eukaryotic cells. Permeases are the membrane proteins which increases the membrane permeability. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Cell membrane is permeable to CO, glutamic acid, and glucose, but not for glucose 6-phosphate. Thoroughly washed beetroot slice kept in water room at room temperature does not lose any anthocyanin pigment because cell membrane is impermeable to anthocyanin. Many molecules can move briefly across the membrane without any requirement of energy, and this is called a passive transport. Neutral solids may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to low concentration. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. Some of the integral proteins run throughout the lipid bilayer and is called as transmembrane protein. Transmembrane proteins act as a channel or carrier proteins. A few ions of or molecules are transported across the membrane against the concentration gradient, that is from lower to the higher concentration gradient. Such a transport is an energy-dependent process. It is in which ATP is utilized and is called active transport. Na and potassium pump, sodium and potassium pump. Besides the active and passive transport, another method used to transport the substances across the cell membrane is called as a bulk transport. Bulk transport occurs in two methods: pinocytosis and phagocytosis. Pinocytosis, term Lewis. Pinocytosis is also called as cell wrecking. It is bulk transport of fluid matter and other substances like iron, sugars dissolved in it. Phagocytosis, term Machenschhoff. It is also called cell eating. Phagocytosis was first seen by Machenschhoff. Phagocytosis by WBC is an important defense mechanism of the body, neutrophils and monocytes. Cytoplasm. Term cytoplasm was coined by Strasburger. Fifth, cytoplasm is the main arena of cellular activities in both plants and animal cells. Cell organelles of eukaryotic cells are embedded in the cytoplasm. Cytoplasm shows continuous movements inside the cell called cytoplasmic streaming or cyclosis. Cytoplasmic streaming helps in the transport of various substances into the cell. Cyclosis is of two types: rotation and circulation. In rotation, the cytoplasm moves in one direction around the vacuole, example young leaves in hydrilla. In circulation, the cytoplasm moves in different directions, staminal hair or trans. Transit cantia or royo, endomembrane system. While each of the membranous organelles is distinct in terms of its structure and functions, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes the endoplasmic reticulum, complex lysosomes, and vacuoles. Since the functions of the mitochondria, chloroplasts, and merosomes are not coordinated with the above compartments, these are not considered as a part of the endomembrane system. Endoplasmic reticulum. The ER discovered and named by Potter et al. Term lipochondria was suggested for ER. ER is a membrane system considered to be extension of infolded plasma membrane. Electron microscopic studies of the eukaryotic cells reveals the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm that is called as endoplasmic reticulum ER. Hence, ER divides the intercellular space into two distinct compartments: luminal inside ER and extraluminal cytoplasm compartments. ER is a single membrane bound cell organelle. Fifth, the ER shows ribosomes attached to the outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called as rough endoplasmic reticulum (RER). In the absence of ribosomes, they appeared smooth and are called as smooth endoplasmic reticulum (SER) versus RER is frequently observed in the cell, actively involved in protein synthesis and secretion. RER is well developed in cell engaged synthesis of secretory product. They are extensive and continuous with the outer membrane of the nucleus. Nucleus granules are formed of RER and dRNA. The smooth endoplasmic reticulum is the major site of synthesis of lipid and steroids. In animal cells, lipid-like steroidal hormones are synthesized in SCR. SCR is also involved in the synthesis of glycogen, cholesterol, fat, and oil. SCR takes place in detoxification of liver. Detoxification of lipid-soluble drugs and other harmful compounds in ER is carried out by cytochrome P450. ER involves in glycosylation, polypeptide modification, and lipoprotein synthesis. ER provides mechanical support to the cell, so it's called endoskeleton of the cell. ER is more developed in mature cells and less developed in rapidly growing cells. ER are quite extensive in metabolically active cells like the cells of the liver and pancreas. ER absent in human RBCs, eggs, and prokaryotic cells. ER of muscle fiber is called cytoplasmic reticulum. So basophilic ergostoplasm, granular ER of glands show richness of ribosomes. Golgi apparatus. Camilo Golgi first observed densely strained reticular structures near the nucleus. GB is Golgi body is present in perinuclear area. These were later named as Golgi bodies after him. Golgi apparatus or apparatus reticulare or Golgi complex or Golgi body (GB) are called dictyosomes in plant cells. GB absent in prokaryotes, bacteria, and BGA, while absent present in yeast. GB is a single membrane bound structure, namely GB develops from ER. They consist of many flat membranous disc-shaped bags or cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. A varied number of cisternae are present in Golgi complex. The functional unit of GB is cisternae. Cytoplasm around GB shows absence of cell organelles. This area is called zone of exclusion. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex, cis or transforming phase and concave trans or maturing phase. The cis and trans phases of the organelles entirely are different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intercellular targets or secreted outside the cell. Materials to be packed in the form of vesicles from ER fuse with the cis phase of the Golgi apparatus and moves toward the maturity phase. This explains why the Golgi apparatus remains close association with the endoplasmic reticulum. The main function of GB is secretion rather than the storage. A acrosome of animal sperm is formed by GB. GB involved in root hair formation. GB involved in transformation of cell membrane. GB produces both lysozyme and vesicles. Hypotoxin of nematoblast is formed by GB. GB secretes mucin in the respiratory tract and slime in insectivorous plants. GB is the export house of cells. A number of proteins synthesized by ribosomes on endoplasmic reticulum are modified in the cisternae of the Golgi apparatus. Therefore, they are released in the form from its trans phase. Golgi complex is specialized for the glycosidation or glycosylation of lipids and proteins. Golgi apparatus is the important site of formation of glycoproteins and glycolipids by glycosylation. Final step of glycosylation occurs in GB, while the initial steps are taken in the ER. The glycoproteins and glycolipids are biochemicals that involved in cell recognition and addition. Lysosomes were discovered by C.D. Doof. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. Hydrolysis optimally active at the acylic pH. Acid phosphatase is present in lysozyme. These enzymes are also called acidic hydrolysis. These enzymes are capable of digesting the micromolecules, carbohydrates, proteins, lipids, and nucleic acids. Lysozymes are also called suicidal bags due to the presence of digestive enzyme in them. Lysozymes are present in animals and cells except mammalian RBCs. Lysozymes are absent in plants. Lysozyme formation is done by both GB and ER. So GERL, Golgi body, endoplasmic reticulum, related lysosome is related to lysosome and associated with GB. Lysozymes are present in all plant cells except mammalian RBCs. Lysozymes are absent in plants. Lysozyme formation is done by both GB and ERL, so GERL. If lysozymes get ruptured in cell, the cell dies. Autolysis. Lysozymes reduce the number of other organelles. Disappearance of tadpole tail during metamorphosis is brought about by lysozymes. Lysozymes are of four types: primary, secondary, autophagosomes, tertiary lysosomes. So it is called as polymorphic cell organelle. Lysozymes are involved in both intracellular and extracellular digestion. Osteoclasts release the enzymes to digest the bone. Fourth, vacuoles. Vacuoles is a non-living cell inclusion. The vacuole is a membrane-bound space found in the cytoplasm. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can be occupied 90% of the volume of the cell. It contains water-dissolved substances, sap, excretory products, and other materials not useful for the cell. In the vacuole, some water-soluble pigments like anthocyanin and anthocyanin are present, which gives red color to rose petals. Colors of petals are due to anthocyanin. In plants, the tonoplast facilitates the transport of a number of ions and other materials against the concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. Vacuole in the plant cell helps in the storage, waste disposal, and cell elongation and protection. In amoeba, the contractile vacuole is important for excretion, osmoregulation. In many cells, as in protists, food vacuoles are performed by engulfing the food particles. Mitochondria, discovered by Collier and named by Banda. Mitochondria stained by Janus Green. Mitochondria involves in Krebs cycle and oxidative phosphorylation. Mitochondria is absent in mammalian RBCs, human RBCs, and prokaryotes. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variably depending on the physiological activities of the cell. In chlorella, green algae, only one mitochondria are present. In terms of shape and size, also considerable degree of variability. Observe. Typically, it is sausage shape or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is called as a matrix. The outer membrane forms a continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called as a crista towards the matrix. The outer membrane has proteins channels called porins. Outer membranes resembles a sieve, hence permeable to all kinds of molecules. Mitochondria without an outer membrane is called as mitoplast. The cristae increases the surface area. Cristae are the sites of oxidation reduction reactions or oxidative electron transport. Cristae possesses small tennis racket-like particles called oxysomes or F0 or F0 particles or elementary particles or Fernandez-Moran particles or Rackers particles. Oxysomes occurs on the inner mitochondrial membrane. Each oxysome has a head F1 subunit, a stalk, and a base F0 subunit. Outer and inner membranes of mitochondria are structurally and functionally dissimilar. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration or Krebs cycle. They produce cellular energy in the form of ATP, hence are known as powerhouses of the cell. The matrix also possesses single circular DNA molecules, a few RNA molecules, ribosomes 70S, and the components required for the synthesis of protein. The mitochondria divide by fission. Among all cell organelles like GB, lysozyme, chloroplast, mitochondria, peroxisomes, mitochondria have highest number of enzymes. Mitochondria is rich in Mn, while chloroplast is rich in Mg. Plastids. Term plastid was given by Ernest Haeckel. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscopes, microscopes as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the types of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. These all three have the ability to multiply by a fission-like process. The chloroplast contains chlorophyll and carotenoid pigments, which are responsible for trapping light energy, essential for photosynthesis. Chromoplast. In the chromoplast, fat-soluble carotenoid pigments like carotenes, xanthophylls, and others are present. This gives the part of the plant yellow or orange-red color. The chromoplasts are formed either from chloroplast or leucoplast. The conversion of green tomatoes into red form is due to the formation of chromoplasts from chloroplast. Lycopene is tomato and capsanthin is in red pepper. Chromoplasts are formed from leucoplasts by development of some pigments like carotenes and carrot. Chromoplast provides color to the many flowers. All plastids have similar structures, so they can get transformed from one type to another. Leucoplast. The leucoplasts are the colorless plastids of weight, shape, and size which store nutrients. Amyloplast stores carbohydrates, starch, and example potato. Elaioplast stores oil and fats, whereas the elaioplast stores protein. Potato. Elaioplast stores oil and fats, whereas the elaioplast stores protein. And amyloplast are also there. Third, chloroplast. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 nanometer. In higher plants, the shape of the chloroplast is discoid. The number varies from 1 per cell of Chlamydomonas, a green algae, to 40 to 20 to 40% per cell in the mesophyll. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. The root is underground, non-green part due to absence of chloroplasts. Like mitochondria, the chloroplasts are also double membrane bound of the two, and the inner chloroplast membrane is relatively less permeable. Outer membrane is more permeable due to presence of porins. The space limited by the inner membrane of the chloroplast is called as the stroma. The number of organized flattened and disc-like membranous sacs called the thylakoids are present in the stroma. Term thylakoid was given by Menke. Thylakoids are arranged in stack-like piles of coins called grana, single grana, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. Chloroplasts of algae like grana. Agranal chloroplasts without grana are found in bundle sheet cells of the C4 plant. The membranes of the thylakoid enclose a space called lumen. Lumen interthylakoid space, separated from cytoplasm by three membranes. Thylakoids are the structural and functional elements of the chloroplast. On the inner side of the thylakoid membrane, there are rounded bodies present called chromosomes. Chlorophyll pigments are present in the thylakoids. Each chromosome has 230 chlorophyll molecules. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. Chloroplast is the factory for synthesis of sugars, autotrophic eukaryotes. Protein synthesis takes place in the mitochondria, chloroplast, and cytoplasm. Lipids contains of chloroplast is 20 to 30%. In all, it also contains small double-stranded circular DNA molecules and ribosomes. DNA of chloroplast is devoid of the histone protein and have a fewer bases as compared to the nuclear DNA. The ribosomes of the chloroplast are smaller, 70S, than the cytoplasmic ribosomes, 80S. Chloroplast and the mitochondria have self-fabricating units as they both have DNA and RNA. Extrachromosomal DNA is found in the mitochondria and chloroplast. Mitochondria and chloroplast are semi-autonomous organelles or endosymbionts of cells because they possess their own nucleic DNA molecules, can form some of the required proteins, but for most of the proteins, these are dependent on nuclear DNA and cytoplasmic ribosomes. Do not arise de novo. Have membranes similar to those of bacteria. Mitochondria probably originated from bacteria because both have similar types of circular DNA. Both mitochondria and chloroplasts are involved in energy transformation. Ribosomes is the smallest cell organelle. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Pallard. Ribosomes were first seen by Claude but discovered by George Pallard under EM. Electron microscopes has revealed the presence of ribosomes. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S, while the prokaryotic ribosomes are 70S. Ribosomes are classified by their sedimentary rate. Here S stands for spread work or sedimentation coefficient. It is indirect It indirectly is a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Two subunits of 70S ribosomes are 50S larger subunits and 30S smaller subunits. Two subunits of 80S ribosomes are 60S larger subunit and 40S smaller subunit. The smaller subunits fits over the larger subunit. Binding requires Mg2 plus. Below 0.001 millimole of Mg2 plus, the two subunits of ribosomes dissociates. Ribosomes are site of protein synthesis as they are called as protein factories. Ribosomes are made up of RNA plus proteins. Cytoskeleton, an elaborate network of the filamentous proteinaceous structures present in the cytoplasm, is collectively referred to as cytoskeleton. Cytoskeleton is made up of microtubules and microfilaments. Microfilaments are mainly made up of composed of actin. The cytoskeleton in a cell are involved in the many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella, singular flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movements of either cell and surrounding fluid. Cilia are 5 to 10 micrometer long, more numerous and having sweeping or pendular movements. Flagella are comparatively longer, 150 micrometer, and responsible for the cell movement. The electron microscopic study of a cilium or flagellum shows that they are covered with plasma membrane. Their core is called exoneme, possesses a number of microtubules running parallel to the long axis. Microtubules are discovered by D. Robertis and Franchi. Diameter of microtubules is about 250 angstrom. The exoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonomal microtubules are referred to as 9 plus 2 array. Peripheral microtubules consist of 2 microtubules and made up of protein, dynein, axonal, with ATPase activity. The central tubule are connected by bridges and enclosed by a central sheet which is connected to the one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also called interconnected by linkers. Linkers are made up of proteins, nexin. A number of microtubules in a flagellum, including those sharing 3 protofilaments with each other is 20. Microtubule is made up of 13 protofilaments of tubulin protein. Both the cilium and flagellum emerge from the centriole-like structure called the basal body. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. Prokaryotic and eukaryotic flagella differs in microtubular organization and types of movement. Centrosome and centrioles. Centriole discovered by von Bennett and named by Bovary. Centrosome is the non-membrane bound structure. Centrosome is a non-membrane bound structure. Centriole is absent in mature mammalian RBCs and prokaryotes, plant cells, but present in chlamydomonas. Centrosome is present in higher animals, lower animals, and lower plants, but absent in higher plants. Centriole are capable of duplication. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both of the centrioles in the centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin. Each of the peripheral fibrils is a triplet. The adjacent triplets are also linked. Centriole has 9 peripheral fibrils, but fibrils are absent in the center. The arrangement is therefore called 9 plus 0. The central part of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made up of proteins. The centrioles or microtubules form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus. During cell division in animal cells, microbodies, membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Microbodies are of 2 types, glyphosomes and peroxisomes. Glyphosomes. These are single membrane-bound structure discovered by viewers and present in only plant cells. Glyphosomes are connected with metabolism of fats. Glyphosomes possess enzymes from beta oxidation of fatty acids and glycosylate pathway. Differences between plants and animal cells. Animal cell wall is absent. Glyphosomes convert fat to carbohydrates by glycosylate pathway. Glucogenesis occurs in glyphosomes. Glyphosomes are found in germinating oil seeds like castor and seed. Peroxisomes, single membrane bound organelles discovered and named by C.D. Dubé. These are found in both plants and animal cells. In plants, peroxisomes are found in photosynthetic cells and perform photorespiration. For this, they are associated with chloroplast and mitochondria. Photorespiration occurs in mesophyll cells. Peroxisomes contain enzymes catalase, peroxidase, and glycolic acid oxidase. Glycolate metabolism occurs in peroxisomes. Sterosomes or oleosomes are single membrane bound organelles associated with the storage and synthesis of lipid. Sterosomes arise from ER. Sterosomes occurs abundantly in endosperm cells, oil seeds. Castor. Nucleus as a cell organelle was first described by Robert Brown as early as in 1831. Later, the material of the nucleus stained by the basic dyes was given the name by chromatin by Fleming. Function of cell is controlled by nucleus. Role of nucleus is hereditary was demonstrated by Hamerling on a unicellular nucleate green algae Acetabulata. Hamerling experiments involved exchanging of rhizoid and stalk. Normally, there is only one nucleus per cell. Variation in the number of nuclei are frequently observed. Most of the cells are uninucleate by paramecium have 2 nucleus. Multinucleate or syncytial conditions is found in epidermis or ascaris muscle fiber. Multinucleate or cenostatic conditions is found in phycomycetes, Smucker, Rhizobus albicue, and Washeria. Acellular slime molds have multinucleate protoplasmic body called plasmodium. Some mature cells even lack nucleus, example erythrocytes of many mammals, and sieve tube cells of vascular plant. These cells are enucleated or anucleated. The interface nucleus of cell when it is not dividing has highly extended and elaborate nuclear protein fibers called chromatin nuclear matrix and one of the most plentiful bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by fusion of two membranes. These nuclear pores are the passages through which movements of RNA and protein molecules take place in both directions between the nucleus and cytoplasm. Proteins required for functioning of nucleus are synthesized in cytoplasm. The nuclear matrix or the nuclear plasm contains nucleolus and chromatin. The term nucleoplasm was given by Strasburger. The nucleoli are spherical structures present in the nuclear plasm. The content of nucleolus is continuous with the rest of the nuclear plasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. In eukaryotic cells, the units of ribosomes are synthesized in nucleolus. The nucleolus was given by Baumann. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall the interface nucleus as a loose and indistinct network of nuclear protein fibers called chromatin. But during the different stages of cell division, cells show structured chromosomes in a place of nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also some RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 4623 pairs chromosomes. Chromosomes were discovered by Hofmeister and named by Waldeyer. DNA is found in chromosomes, mitochondria, and plasma. Circular DNA is found in chromosomes, mitochondria, and bacteria. Every chromosome essentially has a primary construction or the centromere on which the sites of disc-shaped structures called kinetochores are present. A centromere is the site of which the sister chromatin are joined. Kinetochore is the surface of the centromere. The shape of a chromosome is determined by the position of centromere. Based on the position of centromeres, the chromosomes can be classified into four types. The metacentric chromosome has medial centromere forming two equal arms of chromosome. Shape of metacentric chromosome is V-shaped. The submetacentric chromosomes has a centromere nearer to the one of the chromosome, resulting into a longer and shorter arm. Short arm of the chromosome is designated as P, whereas long is designated as Q. Submetacentric chromosomes are L-shaped. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and very long arm. In acrocentric chromosomes, centromere is subterminal and capped by telomere. Acrocentric chromosome is J-shaped. The telocentric chromosome has a terminal centromere at one end. Telocentric chromosomes appear rod-shaped. The chromosome which lacks a centromere is called acentric chromosome. So sometimes a few chromosomes have non-spinning secondary constructions at a constant location. This gives the appearance of a small fragment on the satellite or Treban. These chromosomes are called satellite chromosomes. Nucleolus is formed by satellite chromosomes. End of the chromosome is called telomere. Telomeres seal the end of the chromosome. Non-membrane bound cell organelles, ribosomes, centrosomes, centriole, nucleolus, cytoskeleton structures. Single membrane bound organelles, cell organelles, ER, GB, lysozyme, vacuoles, microbodies, glycosomes, and peroxisomes. Double membrane bound cells, organelles, plastid, mitochondria, and nucleolus. Giant chromosomes, polytene chromosomes, and Lamb-Rush chromosome or are giant chromosomes formed due to duplication without separation. Polytene chromosomes, salivary gland chromosomes. Polytene chromosomes were first discovered by Balbini in Chironomus. Polytene chromosomes is found in the salivary glands of some insects or Drosophila or Chironomus, so it is called a salivary gland chromosome. Polytene chromosomes have swelling on their surfaces which are called as Balbini rings, puffs. Balbini rings are the sites of RNA and protein synthesis. Puffs are specially concentrated with RNA polymerase. In polytene chromosomes, pairing is formed due to homologous chromosomes. Point at which polytene chromosomes are attached is called as chromocenter. Lamb-Rush chromosome, discovered by Rueckert. Lamb-Rush chromosomes are diploiding chromosomes are found in diploiding stage of most animals, oocytes of frogs or amphibians. Lamb-Rush chromosomes are observed in the meiotic prophase. Lamb-Rush chromosomes resemble diploidine chromosomes and are in permanent diploiding stage. Lamb-Rush chromosomes are larger than polytene chromosomes. These chromosomes show chiasmata.